In this video, we're going to have a look at histology. Now, histology drives many students crazy because when you look down the microscope, it just looks like a hot mess. So let's take a few minutes. Let's define what's histology, talk a little bit about how we prepare the slides, because that's really important for understanding what we're going to see. And then let's look at epithelial tissue. Look at the classifications. When you're looking at histology, histological sections, tissues, you really want to have a checklist in your brain. So let's run through a checklist for epithelium, and also let's look at some examples. So let's head in there. Now, the first thing we have to know is that when we're looking at tissues, they've been highly manipulated. Now, hopefully we've done it, that it's retained its architecture, and what it should look like under the microscope should be equivalent to what it looks like in the body. Now, first thing we have to do is, you just can't take tissue, cut it. You put it on a microscope slide. First of all, it's probably going to be too thick, and the tissue is going to basically rot. So first thing you have to do is you have to fix it. So you're going to use some chemical solutions, maybe even formaldehyde, and you're going to fix the tissue so it doesn't decay. Next thing is you're going to have to cut it thin because the light has to pass through it in a microscope. Now, that presents a challenge because tissue will be mushy. Most tissues will. So what we have to do is we have to go through a long process, multi-day process of slowly removing the water, replacing it with alcohol. Then the alcohol gets displaced. A number of series of baths with wax in them, more wax in each series. And slowly that will put the wax in there and make a solid chunk that the tissue is now embedded in. Now, why do we do it slowly? Because we don't want to rush the process, or else cells will shrink, the tissue will distort. So we do it very carefully so we can retain the same look to it. Now that we have a chunk of tissue, next thing we have to do is slice it thin. So we have to use a microtome. That's a fancy pants name for the knife that we use to cut it, and we cut it super thin. It's got to be thin enough that light can pass through it. Now, before you even do that, we've done all this stuff, you have to stain it. So we're going to use different stains. Now, there are a whole bunch of other different stains out there. Most of them will make things look pinkish and purplish. H&E stain. There's many other stains out there. So don't always rely upon the color that you see to tell you what it is, because you can have the same tissue, use two different stains, and they are different colors, right? And of course, we can do this, and we could be looking at either tissue through light microscopy, where I'm going to use the color dyes. But let's not forget, light microscopy has a limit. You can only go so far in terms of magnification before what we call resolution. Resolution is the ability to distinguish two separate dots with clarity as being two separate dots. And at a certain point, you lose the resolution, and basically it's hard to tell what you're looking at. And it depends on the quality of your microscopes, whether you're using a special lens like oil immersion lens. You're looking at somewhere around 1,000 times where the images are starting to degrade a bit. So then the other options would be the electron microscope. Now, in electron microscope, the preparation is a bit different for the tissue, and what we're going to use is not colored stains. We're going to use basically heavy metals to coat the outside. And here's just an example of a light microscopy, and you can see individual cells, but we can't see inside the cells, and really get clarity of what is in there. But you can see the overall architecture and structure of the tissue with light microscopy. Now here's an EM. And you can see that we can really go to a higher magnification and get excellent resolution. As you can see here, like here we even see mitochondria, we have cell membranes. You can really see detail. Now, in general, most of you will be doing light microscopy. So let's start into tissues. Now, there are 4 basic groups of tissues, and each group has subtypes. But the big main groups are epithelial, and each one of these has its own characteristics: connective, muscle, and nervous tissue. So those are the 4 big groups that we're dealing with. Now, one thing I should introduce is the concept that we're looking at a tissue, its cells and matrix. So when we're trying to identify a tissue, it's a really good idea to have a checklist as to what type of cells I should see. Are the cells tightly packed? or are they loosely spread apart? Now, what is matrix? I think you have an idea what cells are. Matrix is all the stuff on the outside of a cell. So if we had a cell, right, the matrix would be all the stuff on the outside and between the cells. And what's in the matrix? It varies from tissue to tissue. In like a lot of connective tissues, we will see different fibers in there. So really when we're looking at a tissue, it's, hey, what are the cells? How are they arranged? Tightly packed, spread apart. What's in the matrix? Is there a little bit of matrix? A whole lot of matrix? Mostly matrix? These are all the things we have to keep in mind. Let's go right into epithelial tissue. So what is epithelial tissue? By the way, often it's also called epithelium, epithelia, okay? What we see here are the thing that really is going to strike you, and this is going to be the big clue for you that you're looking at epithelium, is the cells are packed together. You really do not see matrix. There can be a little matrix, but you really don't see it. So it's all cells packed together. The other thing is where do I find this? Now, we're not going to go through every organ, but here's a general statement. Epithelial tissue faces the outside world. Example, your skin. Or it lines passages or cavities that face into something. So epithelium is really about facing the outside world or facing into a space. Now, that's going to be one of the big clues for us as we go through here. Now, when we look at epithelium, we can break it into a couple of categories. One is covering epithelial and lining. So it covers external surfaces. It also lines internal surfaces. But there's a second group of epithelial, and these are the glands. So they become part of the secretory tissue in glands, like salivary glands. And we'll have a look at those in a separate video. Now, structure and function go hand to hand in the world of biology. So what are the functions? Well, if you think about it, if you're a tissue that faces the outside world or faces into a cavity, then you're probably about protection, absorption, filtration, excretion, secretion, and also sensory reception. And every epithelial tissue does some or all or most of these. Now, let's look at some more characteristics when it comes to epithelial tissue. There's 5 big characteristics, and we're going to walk through each one, but let's introduce what they are. Number 1 is polarity. Is the top and the bottom of the tissue are quite different. Next thing is, because these are tissues that are stuck together, there's no real matrix between them, they have specialized contacts. So things that we've seen before, desmosomes, stuff like that. We'll have a look at that. And another thing is epithelium is supported by a different tissue type. So underneath epithelium, remember, it faces the outside world and into cavities, but deep to epithelial tissue is connective tissue, which looks very different. Next thing is it's avascular. Now remember what avascular, as soon as you put the A there, it means none. So there's no blood vessels through there. Because look at this is epithelial tissue right here. You don't have room. There's no room for blood vessels, and we'll have a look at that in a minute. But it is innervated, so especially the lower levels can have nerve innervation. Now, when you are a tissue that faces the outside world or into a cavity, it is a rough and tumble existence, my friends. Drying out, facing the environment, chemicals, things moving through a tube that you're lining. It's a rough tumble life, and you got a short life expectancy as epithelial tissue, especially the cells that are really facing the outside world or into the center of a tube or a hollow organ. And that means you're going to have to regenerate. So unlike some of the other tissues, they have really good ability to regenerate. So let's have a look. What do I mean by polarity? Is that they have a top and a bottom that we can identify, and we're going to give those fancy names. So we're going to call one surface the apical. So the apical surface is going to be the upper surface, the part that is exposed to the outside world or into a cavity. Now this, my friends, right down here, this is epithelium. So this is the thickness. This is one of the types of epithelium. You can see how many cells make this one up. And The apical surface is up here because it faces the outside world. And the second surface we can talk about is the basilar surface, and that is down here. So we have polarity. We have the apical surface up here, and we have the basilar surface down here. All right. Now, I got to introduce one more word. Sorry about the terminology, but hey, it's like we're learning a new language because medicine and science has very specific language. And the next thing we want to look at is this, and I'm going to zoom in, is that below the basilar surface, we have something called the basal lamina. Let's look at this. Basilar for bottom. Lamina, to laminate something, means to put it on, layers, attachment, right? So the basal lamina is this bit of tissue right down here at the bottom. Now remember, this is epithelial, and if you remember from our discussions before, what is underneath epithelial? It's connective tissue. So between the epithelial tissue and the connective tissue, we're going to have this thing called the basal lamina. And this is an adhesive sheet. So your epithelium just doesn't slough off as you're walking around doing stuff. Very handy, my friends. Okay. Next thing is, as I said, it has specialized contacts. And these specialized contacts help it to do its job. Because if you're facing the outside world, I don't want stuff to just sort of squeegee its way between cells, because I'm there partly as a barrier. So what we tend to see are things like tight junctions and desmosomes. And if you're not sure what they are, previous videos talk about them, or just look it up in a book or check it out. Tight junctions basically prevent things from squeaking between cells, and desmosomes keep cells from pulling apart, so they really hold stuff together. All right, and so those are some of the specialized contacts we see. Now, the other thing I said is epithelium is supported by connective tissue, and there's some connective tissue we should look at. And two of the terms we're going to see are reticular lamina, okay, and that's deep to the basal lamina. And what is this? This is made of collagen fibers. The next one is the basement membrane, right, and that's the basilar reticular lamina together. And this helps to reinforce epithelial sheets, and it prevents stretching and tearing, and also defines it. So let's have a look at the diagram down here. Here's the artist's diagram of epithelium right down here. Up here, I should say. Down here, it's connective tissue. So remember, what is below epithelium? Connective tissue. And in between, we see that we have two different layers. The basal lamina right there, and deep to the basal lamina is the reticular lamina. So, basilar lamina, reticular lamina, together we can just refer to them as basically the basement membrane. Okay, so that's some more of the features. Now, the other thing I said is it's avascular. Now let's have a look. This is a type of epithelial tissue here, which we'll have a look at shortly. So this is connective tissue down here. You can see that, once again, one of the features of epithelium, it's all cells. No matrix. There is no room for blood vessels. It is avascular. So if you're wondering, these are living cells. Where does it get its nutrients from? Diffusion from the underlying connective tissue. All right, and the next thing is regeneration. They have the ability to regenerate. So we tend to lose cells up here, and down the basilar area we will replace them. All right, now let's get into epithelium. Now, epithelium, there's a classification system for names. All epithelial tissue has two names. It has a first name and a second name. The first name tells us how many layers thick is it. Okay? So if you're only one cell layer thick, you are a simple epithelium. If you are two or more layers thick, you are stratified. So the first name of epithelial tissue will always be either simple or stratified, with one exception, which we'll get to. Right? The second name tells us the shape. All right? Is it flat like a pancake? In that case, if it's flattened and scale-like, we're going to call it squamous or squamous, either one. Or if the cells are now shaped like a cube, we're going to call it cuboidal. And if the cells are taller than they are wide, we are going to call it columnar. Now let's have a look here. All right, so remember, there's two names, a first and a second name. So if I'm simple, I am only one cell layer thick. So let's have a look. So here is a simple type of epithelium, and we can see the cells are fairly flat. So we're going to call this simple squamous. Simple cuboidal, because the cells are cube shaped. And of course, the third one is simple columnar, where we can see the cells are taller than they are wider. Let's go to the stratified. Stratified means more than two or more cell layers. And here we have stratified squamous. Now, let's stop for a second. Because if you look here in this RS diagram, the cell shape down here is a bit different than the cell shape here. Remember, there's an apical and a basilar surface. We always use the cell shape at the apical surface to determine the shape. So these could be cuboidal down here, but no, it's the ones up here that are flat. So this is going to be a stratified squamous, and that's what we see here, stratified squamous, many layers. Here we have a two-layered epithelium, and here the cells are kind of cuboidal. You know, they're sort of cuboidal. They're not as flat as up here. So this would be a stratified cuboidal, so stratified cuboidal epithelium. And down here we have two layers, so we know we're talking stratified, and because the outer layer, the apical surface, they are columnar, this is a stratified columnar epithelium. Now there is one that I should mention, and this one is called pseudo-stratified. Now, why pseudo-stratified? Remember, pseudo means false. It looks stratified, but technically it isn't. So if you kind of squint, you can see these nuclei look like they're all on different levels. And I'm going to show you some histology, so don't run away. We're going to look at this in actual slides, and we're going to perfect our skills. So when you look under a microscope, easy peasy, my friends. Now, what is about pseudostratified? Pseudostratified, all the cells are touching the basement membrane. Yeah, the nuclei may be at different heights, but they're all touching the basement membrane. So it is pseudostratified. Now that we've done that, let's practice. This is really important that we get our eyes used to looking at this. Now, here we are. We're looking down at a slide. I'm zoomed in, and at first, don't get overwhelmed. It is so easy when you first start this to just go, wow, that looks like a hot mess. But let's go back to our criteria. So I got to find some epithelium on here. And remember, epithelium faces the outside world or space. So when I look, I can start to see some spaces. So this is where I would start to really focus in on. And let's start here. This is a big structure. By the way, this is a glomerulus from the kidney. This is kidney tissue we're looking at. You wouldn't probably know this unless you've done a lot of histology, so don't worry about it too much. Now, let's have a look. Because remember, epithelium lines spaces or faces the outside world. So we're going to look in this zone right here. And when we start to look, I'm going to zoom in. You can see, now these are not cells, these are the nuclei of cells. So we can't quite see the cell outline. You can kind of see a bit of a cell outline there, etc. What do you think this would be? Now I don't see other nuclei in this tissue. So this is telling me how many layers? How many layers do you think? One layer. So if it's one layer, it is simple. Now we got to figure out the second name, which is based on cell shape. And The cells look quite flattened. The nuclei are quite flattened. The cells are flattened. So this would be simple squamous epithelium. Right? So there we go. Now, there's a second type of epithelium here. Have a look around for a second. See if you can see it. Remember, we're looking for a space. So here's a space. You can see that there's a very similar pattern, because what we're looking at here are tubes that have been cut across. Imagine taking a garden hose, sectioning it, and looking at the cut end of the garden hose. That is what we're looking at here. These are tubes where we've just cut them while preparing the tissue. And yes, we do have a space here. And if you look here, you can see the various cells. Now, they're not flattened. They're kind of about as tall as they are wide. So, and there's only one cell layer. I don't see multiple cell layers. So this would be simple cuboidal. So now we've seen two. Let's practice a bit more. Let's go here. Now I've told you what it is up here already. This is simple columnar. And let's have a look. Now when I'm looking around, I can see it faces the outside world. I can see the cells are all really packed together. By the way, remember, these are the nuclei. That's not the cell. So the cells would be akin to this. When you start to look, you can start to see the patterns. And this is a simple columnar. It's one layer thick. The cells are column-like, simple columnar. And of course, we know if that's simple columnar, down here, we're probably starting to look at connective tissue. And of course, what separates them would be that basement membrane. And let's try another one. Now, this is the tricky one. This is the one that can throw you off. And luckily, it doesn't show up in many parts of the body. And this is the pseudo-stratified columnar. Now, at first, when you look at this, here's all the nuclei. You would think these are all different cells, but no, they are different cells, but you'd think they're all in different levels. But no, every one of these cells touches the basement membrane. So remember, technically, they're not stratified. This is pseudo-stratified columnar epithelium. And of course, what would this be? What's always underneath epithelium? CT for connective tissue. It's connective tissue. Hopefully you're getting the hang of this. And now, so we looked at simple. Let's go into stratified. Now, stratified is more than one layer thick. And here we go. This is stratified. This is way more than one layer thick. We can zoom in. We can see all these cell layers all the way up here. By the way, also look, if you start to look at the cells, they do change shape as we head up. And when we get up here, you can see the cells are really starting to flatten out, especially the ones at the very end. They're really flattened cells. So we already know this is more than one cell layer thick, so we're going to call it stratified. Now remember what our rule is. These look a bit more cuboidal at the basilar surface. We want to look at the apical surface. And at the apical surface, these are a squamous type. So we always name stratified epithelium based on the cell shape at the apical surface. So this is stratified squamous epithelium. Now let's just zoom in. And we'll just move through the thickness of the tissue. You can see that the cells up here on the apical surface much more flattened out, and as we head down, they're a bit more rounded, and they look different. And this is this is one of the major stratified epitheliums that we're looking at here. And down here, they're much more cuboidal. And not surprisingly, the cells at this surface get sloughed off. New cells are made down in this zone, and the cells get pushed up, and they move their way over time here, and they transform. And we'll talk about that transformation when we get to things like skin, etc. Now, you already know this. If this was epithelium, what's down here? This is connective tissue, and that's a whole other video as to how we identify connective tissue. Now, let's have a look at this one, and this is a stratified cuboidal, and it looks like it. You're hopefully getting good at this by now, and I think you are, is that we have a cell here, a cell there. You can see that it's more than one cell layer thick. The cells are pretty cube-like, and so this is a stratified cuboidal that we are looking at right here. Well done. And here's another weird one. This is one of those odd ones. Remember we had pseudo-stratified? This one's called transitional epithelium. Now it is a number of cell layers thick. Why is it called transitional? Because it can transition from one shape to the next. So we see this in very specific parts of the body. I'm not going to go into great detail, but one is the bladder, where the bladder expands and we start to see basically the bladder stretch. So the tissue stretches with it, so it can transition its shape. But most of the times when we see it, of course, it's a histological section where it's not in a bladder full of urine being stretched, so it looks like this. Those are the major types of epithelium. So it's a quick walkthrough. Let's just review. We talked about what tissues are, talked about cells versus matrix. We talked about, hey, how do I prepare tissues? Because we beat them up a bit, but we try to retain the structure. And by the way, sometimes we goof and we make things called artifacts where maybe the tissue rips. So sometimes we end up with fake looking stuff in there, but with trained eye, we'll pick it up. And then we looked at the different ways we name epithelium. Epithelium always has two names, a first name and a last name. First name tells us how many layers, so it's either simple or stratified. And then the second name is based on the cell shape at the apical surface. If you're flat cells up there, squamous or squamous. If you're kind of cube-like, you're cuboidal. And if you're more like a column, you're columnar. That's it. Histology isn't bad, and let me give you the last big, major, helpful tidbit. In the world of histology, they called it like they saw it when they were coming up with names. So look at the names. They're a bit of a cheat sheet for you. So thanks. Hey, if you like this, drop me a comment. If you want more of these, let me know. And feel free to hit the subscribe button. And let's carry on with our journey. And practice, practice, practice. Okay, we'll talk to you soon. Thanks.